So today I'm going to talk about my collaborative project on developing a baseline metabolomic signature that are associated with COVID-19 severity. So Merida has already introduced a little bit about myself. I'm a Ph.D. in progress in my fifth year, and my research interest mainly lies in my multi-omic study, including integration, biomarker identification, and also by clustering for subtyping. So this is one of the by clustering project that I'm working on. I'm trying to find patterns from the map here to reorganize it into detectable patterns like that. So going back to this project, we are already very familiar with COVID-19, and want to emphasize here that although the vaccination has made COVID-19 much more manageable, it still can be a deadly disease. So in August and early September, the CDC reports that every week there are approximately 1,000 people died from COVID-19. And so it's still important to know which patients are going to develop severe COVID outcome at an early stage to prevent them from being from more severe outcomes, including that. And so here's an overview of our analysis. We used the data from two randomized clinical trials, multi-center trials in April and from April to November 2020, across 13 centers in the U.S. We recruited 133 patients. All of them tested positive before their enrollment in our study. And out of these 133 patients, we collected 107 blood samples from them and at baseline, and analyzed their blood samples with MXP called 500 kit by biocrates to obtain 630 metabolites and 264 aggregation. And in our study, we focused on the metabolites because the aggregation is more difficult to interpret. And these metabolites come from 26 compound classes. In the end, we are able to find 3 metabolite signatures. Well, 3 metabolites are shown here. So one of them is from lysopc family, one of them is from dihydroceramide family, and the other one is from the triglyceride family. And so our statistical analysis includes 3 parts. The first part is the univariate analysis. It's mostly served as a preliminary analysis. And the multivariate analysis is a key part of our analysis, where we want to identify signatures that can predict the patient severity of COVID-19 at baseline. We use the resampling techniques to improve our robustness by splitting the data into training and testing sets. We did that for 100 times. And partially square discrimination analysis, PLSDA, was used to find important biomarkers, and then we use that result to identify the signatures. And the last one is the pathway analysis. We want to understand how the metabolites are related to the pathway, so to better understand the disease. So that's the 3 part of our analysis. And I will show first the univariate analysis results. We compare each individual metabolite for hospitalized patients versus never hospitalized patients Oh, and sorry, I forgot to mention the main primary outcome for our analysis is whether or not the patient will be hospitalized during their COVID-19. Yeah, so here we compare each individual metabolite on hospitalized patients versus never hospitalized patients. So the x-axis is the log2-fold difference between those two groups, and the y-axis is whether their difference is significant, so negative log 10 of the p-values, and they are colored according to their significance. Because there are so many comparisons, we also applied Benjamin E. Hirschberg adjustment for multiple comparisons. And so for the PLSDA, for the multivariate analysis, we wanted to find metabolites that are at least that are lowly correlated to each other and can predict well. We indeed found 5 of them, and these 5 of them are the how they showed up in the volcano plot. We know that all of them are significant after Benjamin Hirschberg adjustment, not surprisingly, but also not all of them are very high in the y-axis. That's because the higher ones, most of them are correlated with the lyso-PC family. So that's why we only choose this one there, and the other ones, they are moderately correlated at most. So that's our univariate analysis, and then we go to our multivariate analysis. So this plot shows one example of the 5 plots from the PLSDA with the hospitalized patient and the never hospitalized patients are colored in different with orange and blue. And we see from this plot that they show very good separation between them. And the arrows in the middle is the loading plot for the metabolites, showing how much their role is in creating the spores, those either rectangle or circles. And we also see that the right lines, these are the loadings for the 5 metabolites selected from the PLSDA. And next step, we want to create a predictive model using those metabolites. And so here we show the ROC curves of those models. We started from the clinical covariate only model, which includes age, sex, and BMI, and then we sequentially add one more metabolite to the model. And the reason why we only include the first 3 is that when our sample size is a little bit small, only 107 if we include, and we split it into training and testing. So the training sample is only 70, about 77 of them. So once we include more metabolites, the model will diverge, logistic regression model will diverge. So that's why we ended here with only 3 metabolites. And we can see from here, these are the testing AUC. Using the 3 metabolites can show a very good predictions based on the AUC, much better than using the clinical covariate only. And last but not least is the pathway analysis. So these 5 are the 5 highly enriched pathways we identified in our study. These are glycine biosynthesis, threonine degradation, tRNA charging, phenylalanine degradation 4, and Kaifeng degradation. So the error level lies between shows that shows whether there is shared metabolite between those pathways in our analysis, and the number here are the number of shared metabolites there. So both other pathways are related, have exactly two shared metabolites with the tRNA charging, which is interesting, and we think it might be worthwhile for other researchers to dig in deeper about what role tRNA charging plays in this process. So I want to conclude my presentation with the discussion and limitation of our study. So we are one of the few groups to use metabolomic data to differentiate hospitalized patients from never hospitalized patients. Other studies I found there are studies studying like hospitalized patients on positive versus negative COVID, or like in general positive versus negative patients, but there are only we are one of the few to do the hospitalized versus never hospitalized patients, and we are able to identify a 3 metabolite signature to assist the dichotomizing patients when at baseline. And we also found several pathways to be significantly enriched for future researchers to dig deeper into. And future studies, they can attempt to create a more accurate model, probably with a larger sample size, so that we can include a larger number of metabolites as signature. We see that while we every time we add a metabolite into it, the performance gets better. So we think if we can have a larger sample size with all 5 signatures, the performance might be even better. And another limitation of our project is that it's all the patients are enrolled between April to November in 2020, so it's limited to 1 or at most like a couple early strain of COVID-19. So our finding may not necessarily generalize to the whole COVID-19 group, but other researchers, they can use our results or data to validate their studies and to help generalize it to the more general strains of COVID-19. And that's the end of my presentation. Any questions or comments?